Hello. Hi, Debbie.
Hello.
Hello, everybody. Wednesday. Hi, everybody. Good to see you. Good to see you today.
Hello, you as well.
I've got my tea. I'm all set. Thank you for typing your name into the chat. I appreciate that. And also, if you have a nickname that you want me to know, feel free to type that into chat as well. I forgot to ask that early on. I know some people don't go by their given names. My dogs are trying really hard to get in my lap. We'll see if we can keep them off today. It's funny, they have both learned. I have two female dogs, and they have both learned that when I sit down like this at the table, that I'm going to be talking, and for all they know, talking to my computer for a while. And they're both starting to get a little jealous of it. So every time I come over to sit down, they try to get in my lap.
Do you adjust the scores of the exam at all?
I adjust scores if there's my system, my personal system is, if there's a question that fewer than 25% of the class gets right, I drop that question and I give the points back to everybody.
Okay.
I don't typically make any other adjustments though.
No, I was just wondering because I took my test as you saw, and my score.
Don't forget though. Don't forget, there's at least one question on that exam that I have to grade by hand.
Well, I wasn't pleased. I don't know. It just didn't seem right. I don't know. I thought I did good.
I always go through all of the questions, but remember, on the exams, there's always at least a question or more that are short answer questions, and Canvas can't grade those. I have to grade those. So I always tell students, try not to worry when you hand in the exam when you see a lower score than what you might want, because there's stuff on there I have to grade by hand. And also remember that the lecture exams are out of 80 points, not 100 points. So that's important to remember too, so you don't have a heart attack. All right, looks like everybody's here. So we'll go ahead and get started. I did send just a brief announcement out earlier to remind you that there is a lecture exam to complete today. If you haven't already, I think just about everybody has. And then what we're going to be doing today in lab is talking about the fungi. So this is the one day really that we talk about fungus in all of its glory. I do know we did not get a chance to finish all of the slides in our last laboratory time together. And if you recall, we were talking about colonies and what a colony is, and CFUs. And the last, I think there's maybe 5 or 6 slides we didn't get to that talk about colony morphology in detail. And actually, we look at some colonies growing on a plate. So what I'm going to do with those last 5 or 6 slides is I'm going to make a short video and post it for you. So it'll be between 5 and 10 minutes long, and it'll just be what you need to know from that last few slides. Now, there was a bonus question about that material. Obviously, we hadn't talked about it yet, so I didn't expect everybody to try that bonus question, but several of you did. And I was pleased to see several of you got at least one of those questions correct. So again, stay tuned. If you didn't answer the question, if you didn't try it, or if you didn't get it right, stay tuned for that short video. I'll try to post that for you tomorrow. Just again, finishing up that set of slides that we worked through together last time. So as always, I want to ask first if anybody has any other questions about anything before I get started with today's material. I don't know if anybody's had an opportunity to do much more than take the exam, but we can always talk about any questions you might have during these lab periods too.
Well, I was going to ask about the project.
Oh,
okay.
Are
we doing like a paper or just answering the questions? So
you're just answering those questions. Now there's instructions on the document. In fact, I can pull it up for us. Hold on just a sec. There's instructions on the document that tell you more precisely what I'm asking you to do. Let me pull this up for us. Put this down. Here we go. So you should be seeing our home page on Canvas. I'm going to go ahead and click on modules, and I'm just scrolling down to find the pathogen project, sorry. This project looks different at in different semesters. Sometimes we have students write a paper, and sometimes we do it this way, and sometimes we have students fill out a chart and all kinds of different things. So this semester, what we're doing is letting you choose a set of questions to answer. Okay, so there's 4 groups of questions that you get to read over, and you just have to pick one group of questions that you want to answer about one pathogen. And then it says you have to refer to at least 3 different references for each question in the group that you choose. And after you answer the question, you just want to let me know what references you used. I gave you a list of some good websites. There are plenty more websites out there that are reputable and, based on science and not, just, somebody's very casual website. So down here it says, actually, we'll start here. Of course, everybody has to submit their own assignment written in their own words. I don't want you to quote your source materials directly. In other words, I want these answers to be in your words. So if you want to talk about something that you read from a source, just put it into your own words. Don't put quotations in there with a direct quote. And I also ask that you don't use any scientific jargon in your answers. If you come upon some jargon, in other words, some very technical scientific terms that you don't understand, I don't want you to use those terms. I want you to put it into your own language. And then I described that you're submitting this in 2 stages. You're going to hand in your first draft, essentially, of your answers. And then I'm going to take a look and submit them back to you with recommendations. And then I said, no one answer should be longer than one side of one page. Use complete sentences in your paragraphs, correct spelling and grammar. Okay, and then here's the groups of questions that you are choosing from down here at the bottom. So, again, somewhere in here I say to write your answers in paragraph form. Again, full, complete sentences. But there's no, minimum length of answer or anything. Some of these answers you'll be able to write out in a sentence or 2. And some of them you might need to write a paragraph or 2. All right, but you're just answering a series of questions not writing a formal paper. Okay. Yeah, so hopefully you've taken a look at this and gotten started on it. It is due a week from today. It's due the 24th. Again, in that first draft form. And remember, the first draft you want to put your best work into, because your first draft is worth 35 points. And then after I return it to you and you make revisions, your revised draft is worth 15 points. So in total, you can earn as many as 50 points for this. So it's worth putting a little time and effort into. I'm not fussy about how you cite your references. In other words, let's say you find a journal article, a primary source journal article online, and you use it to answer a question and you have to cite it. You can choose any citation style you want to use, whatever you're comfortable using. Just make sure you provide a complete citation and make sure that you use the same style all the way through your work. So another thing to remember is it's not enough just to tell me that you went to www.cdc.gov. That's an enormous website. You have to tell me where'd you go? What page did you go to? What was the URL that you went to specifically to get your information? Okay. Remember, the reason that you cite references, not only are you telling your reader that these are, I got help with this. These are not all my original thoughts. But you're also telling your reader where they can go to find that material if they want to take a look at it too. So, so yeah, make sure you have a full and complete URL or web address if that's what you're using, or some other citation if you're using some other material. And that's due a week from today. All right, very good. Any other questions? All right, very good. So I'm going to pull up our slide set for today about the mighty fungi. The first thing I want to say about fungus is this is a topic that stands on its own. You can take a semester-long course about the fungi. They are incredibly diverse. They are incredibly complex organisms. The study of fungi is called mycology, and there are people who are experts in it. I am not an expert in fungi. But we recognize certain types of fungus as microbes, which is why we learn about them in this course and why we'll be talking about them today. Our objectives today are to talk a little bit about how we characterize the fungi, how we classify the fungi. It's a lot more complex than the way we do it with other organisms. We're going to compare and contrast the fungi with bacteria. Where are they similar? Where are they different from bacteria? We're going to put a name to some of the common groups of a particular type of fungus called a saprophytic mold, and we'll talk about why they matter. And then we'll finish up talking about some features of, again, microbial, or I should say microscopic fungi, yeasts and molds specifically. So let's review what we know about fungi from the early part of the course, from that introduction to the microbes material. The fungi are considered eukaryotic, so they belong in the domain eukarya. Remember, there's bacteria, there's archaea, and there's eukarya. So all of the eukaryotic organisms are in the eukarya domain. Now, because they're eukaryotes, that means they can be unicellular, they can be multicellular. Remember, there are some microbes in the eukaryotic domain. And then there are lots of other organisms that don't fit the definition of a microbe. Fungi can be microscopic and they can be macroscopic. We see both types. Now, when we think about the microscopic fungi, there are two types. There are yeasts and molds. But only one of those is a microbe. And that's because remember what our definition tells us. A microbe is a single-celled microscopic organism or acellular particle. So the only one of these microscopic fungi that are single-celled is the yeast. Turns out the molds are multicellular, even though they're microscopic. Now, both of these are considered vegetative forms, in other words, cellular forms. Remember, in bacteria, we talked about how you can see vegetative bacterial forms, cellular, and you can also see spores in certain types of bacteria. And we said that those bacterial spores are really in suspended animation. They're not doing anything that living cells do, but they can be brought back to the vegetative form if they're fed water and nutrients and they're in a warm temperature and so on. We're also going to talk about spores in relation to fungi, but, and this is why I have two stars next to this, the spores that we talk about in relation to different types of fungi are not the same as those inanimate spores we see in bacteria, in some kinds of bacteria. When we talk about spores related to fungi, we're talking about reproductive structures. I like to think of them as being similar to like pollen in a plant. So the yeast use spores as their reproductive materials, as their reproductive molecules. Now, those spores are not cells, so they're considered, non-vegetative. The yeasts and the molds are cellular. They're made out of cells, so they're considered vegetative. And both yeasts and molds, as well as these spores, these are all microscopic. You need a microscope to see these. Now, there are, of course, many macroscopic forms of various fungi. You're already aware of that. I'm sure you may or may not eat certain types of fungi in your diet. When it comes to the macroscopic types, the kind you can see with your own eyes, the kind you don't need a microscope to see, we're talking about multicellular forms of fungus. So these would not be considered microbes. There's a vegetative type of macroscopic fungi. There's a cellular type, and that's called Mycelia. Mycelia is a plural word. So one of these would be called a Mycelium. Now, the interesting thing about these vegetative forms of multicellular macroscopic fungi are that they grow underground. So you and I, as we walk around this earth, we are constantly walking on top of mycelia. They're under there and they're growing virtually everywhere. Everywhere that there's dirt, there are mycelia growing. Now, there's another macroscopic form that's vegetative, cellular, and those are mushrooms. So the things that you and I like to eat, these mushrooms, as we call them, those grow above the ground. And what a mushroom really is a reproductive stage of the mycelia that are growing under the ground. So the mycelia send up these mushrooms through the soil and up into the air when they're ready to reproduce. If you've ever encountered, not all mushrooms, but some mushrooms that grow, say, on your lawn or in your garden, sometimes when you step on these mushrooms, they will release a sort of a little cloud of dust. If you've ever seen that happen, those are spores coming out of that mushroom. Those are the reproductive materials of that particular fungus. And the fungus, is trying to make more of itself, trying to reproduce. Now, all of these different forms, the microscopic versus the macroscopic, and single cells versus multiple cells, and growing underground and growing above ground. As I said earlier, the fungi are incredibly diverse. So some of them spend part of their life as microscopic organisms and part as macroscopic, and they are very, very diverse. They have very complex life cycles. One of the interesting things about fungi compared to other organisms and other microbes is that they get their nutrients through absorption. So they literally absorb their nutrients across their cell wall. And remember what their cell wall is made of. Anybody remember? What's the cell wall made of around a fungal cell, around a yeast cell, for example? It's not peptidoglycan.
Is it chitin?
That's right, it's chitin. Just like the material, it's the same material that you find in the exoskeleton of an insect, that crunchy material around an insect. Same stuff in the cell wall of a fungus. So nutrients will pass through chitin and then pass across the membrane to feed those cells. Most of the fungal species that you encounter are actually aerobic. In other words, they use oxygen for metabolic purposes, just like you and I do. But there's a big exception, and that's the yeast. And since the yeast are the only microbial fungi, that's important for us to remember. So some species of yeasts are aerobic, they use oxygen. Some species are anaerobic, so they don't need oxygen to do their metabolic functions. They can use other molecules instead. Yeasts, again, like most fungi, come in all different forms. They're very diverse. So I want to take a couple minutes and compare the fungi with bacteria, particularly in terms of their growth characteristics. So if you look on this slide, the first thing I have written is that when we compare fungi to bacteria, the fungi have a couple of important characteristics. Number one, they can tolerate a lot more acid in their environment, a lot more acidic pH in their environment than the bacteria can. Bacteria are very sensitive to pH. Now, why does that matter? Well, if you think about something like this tomato in the image growing in the garden, and if you're a home gardener, you've probably seen this on your tomatoes from time to time. The fungi can grow on something like a tomato and kill it, or at least make it so we don't want to eat it, whereas bacteria really can't. Bacteria can't because the tomato is too acidic. So think about all of the acidic foods that we eat. Just the nature of the food, the fact that it has an acidic pH, remember that means less than 7. The fact that it has an acidic pH means that bacteria just really can't tolerate it as a food source, but the fungi can. Now, they can only tolerate it to a certain degree. They can tolerate a pH down to the area of maybe 4 or 5, but not much below that. So acidic vegetables, tomatoes a good idea, Another good example is acidic fruit, like this orange down here. You can see it's got a big mat of fungus growing over it. It doesn't look like a pathogenic fungus or anything, but I wouldn't want to eat that orange. Bacteria aren't going to grow on these. Bacteria don't grow on oranges or lemons or limes. It's just too acidic of a food source. But the certain types of fungi, they don't mind the acid at all. And that's why if you leave the oranges in your fridge too long, they often grow fungus on them. So compared to the bacteria, the fungi can tolerate a lot more acidic pH than the bacteria can. They can also tolerate less water. Now, this is especially important when it comes to food spoiling. It turns out that bacteria have a very set, very real need for a certain minimum amount of water in order to grow and survive. The fungi are able to tolerate less water in food when they grow. Now, where we really see this in our own food supply is in sweet things. So I'm talking about things like, again, fruit. Jams and jellies are a really good example. And an especially good example is syrup. So here in New Hampshire, where we love our maple syrup, you don't get bacteria growing in these types of foods. And the reason you don't is because there's not enough water in them. Now, you might say to yourself, well, hold on a minute. The last time I cut into an orange, the orange was very wet, very juicy. Or the last time I cut into an apple, it was very juicy and delicious. And my strawberry jelly this morning on my bread was certainly wet. It has water in it. My maple syrup certainly has water in it. Here's the thing, though. These sweet things also have a lot of sugar in them. They have carbohydrate, which is all sugar is. We use the word sugar, but technically speaking, we're talking about carbohydrate. And carbohydrate has a tendency to hold on to water. It binds to the water. So yes, there is water in your strawberry jelly, but it is technically bound up to the sugar in the jelly, to the fructose or the sucrose in the jelly, and it's not available for the bacteria. It's especially apparent in things like syrup. So, when you buy maple syrup, it'll say on the container that once you open it, you need to keep it in the fridge. But the truth of the matter is, bacteria just can't grow in maple syrup, but fungi can. So a fungus can grow much more easily on fruit, in jams and jellies, and in syrups. They tend to grow on the top. They tend to grow on the surface. So if you've ever left a jar of strawberry jam in the fridge for too long, again, and you open it up one day and you find some fuzzy green fungus growing on it, it's not that unusual. And the same thing happens with syrup. You can find fungus growing on the surface of syrup relatively commonly. You won't find bacteria, though, because there's not enough available water. The water that's in these foods is being bound up and held onto by the carbohydrate in the food. Very fascinating, and an important thing to know about food safety. Now, let's turn it around. And instead of comparing the fungi to bacteria, let's compare the bacteria to fungi, and where do they stand? Compared to fungal cells, bacterial cells can tolerate shifts in osmotic pressure much better. So it turns out that the chitin in the cell wall of a fungal cell is not nearly as protective as the peptidoglycan is in a bacterial cell wall. And this is especially evident when it comes to osmosis. bacteria are protected against big shifts in osmosis, in osmotic pressure. Now, we talked about osmosis as a you know a phenomenon, and we said that osmosis can drive water into a cell or out of a cell, and it can kill a cell if it happens too quickly and at too great of a degree. So you can imagine if you're a single-celled organism living in the world, you would be very vulnerable to finding yourself in an environment where there'd be a big osmotic shift. Bacteria are protected against this, not 100%, but they're protected against this much more than fungal cells are. So the fungi are more vulnerable to osmotic pressures and osmotic shifts that could end up killing them than the bacteria are. Just a different organism and a different characteristic for that organism. Hold on just one sec. I have a visual for us today. I picked this up at the market basket recently. Some delicious fig jam. And I noticed after I took the plastic wrap off and I went to open it, I noticed that the seal was broken. So it didn't pop when I opened it. So I wouldn't have eaten this anyway. But when I opened it up, look what I found. That's fungus. There's a layer of fungus growing right on the top of that jam.
You're going to return it and get your money back?
I was going to, but then I decided it would make a much better visual.
So much more valuable in that regard. You hit the lottery.
You just never know, right? But it's a good reminder that when you get a jarred food that is jarred it should be under a vacuum. You should hear that nice pop when you break the seal. If you don't, it tells you that there's at least a chance that the food inside got contaminated with something. And I knew right away what my thing got contaminated with. All right, very good. Now, I'm not going to talk a lot about the taxonomy or the classifications of different types of fungi, but there are a couple of specific genuses that I want us to be familiar with because they really have a practical application. So just remember that there's an incredible number of species of fungus. They can be very different from each other. They're very diverse. They are genetically diverse. They are morphologically diverse. In other words, they look very different from each other. And they have a tremendous diversity in their life cycles, all the different forms they take during their life. But there are a couple that are interesting historically and have, again, practical application in our lives. The one that has some historical significance is this one. This is the genus name of a type of fungus that we used to think, or I should say, we used to think was a fungus, and we used to call it water mold. So this terrible looking word is pronounced mycota. It looks like it should be mycota, but we pronounce it mycota. And we used to think that this was a fungal organism, and we used to call it a water mold. I'll show you a picture of it in a minute. You can see why we were fooled. In recent years, we've been able to do a genetic analysis of this organism, and it turns out it's really not a fungus. It's much closer to an algae. So genetically speaking, it's much closer to an algae than it is to a fungus. The reason it's important historically is that this is the organism that was responsible for the famous famine in Ireland that drove so many migrants over to the United States in the early parts of the 20th century. if you're not familiar with this history, potato was a very important foodstuff in Ireland at that time, and there was a terrible blight, killing virtually every potato in Ireland, and it lasted for several years. When I use the word blight, what I mean is the potato became visibly spoiled. And again, we blamed it on, well, we know it was this water mold. We know it was this organism, but we always thought it was a fungus. And it turns out it's not. It's its own type of organism. This is a series of pictures of Oomycota taken these are both grossly visible structures and also structures that you only see under the microscope from this organism. I think you can see why people thought it was a fungus. It just has that sort of fuzzy look that we tend to associate with fungi, especially like if you look down here, it looks very fuzzy and indistinct. It looks like it's got all these different sort of branched structures. And again, we believed that this thing was a fungus, and it turns out it's really not. And on this slide, what you're looking at is this blight that played such a huge historical role in the history of Ireland. These are this is a potato plant, and these are what the leaves look like when this organism starts to grow on them. They just die, they turn this brown color, they dry up, and they can no longer do their job, which is photosynthesis. And if the plant can't perform photosynthesis, it cannot grow. And then when people would cut open the potato, they would see this terrible ring of black inside and sort of a white powdery material on the outside. Again, you can see why they thought it was a fungus. The potatoes were spoiled. They were not good for eating. And because they were such a staple in people's diets at the time, there was terrible starvation in Ireland. And
may I ask though, is the inside of the potato not salvageable at that point once it has the blight?
yeah, I don't know enough about it historically. Obviously, those pictures were taken in more modern times, but I'm not sure historically if people were able to sort of carve out a little bit of the potato, maybe harvest them a little early. I have seen pictures of what happens when that potato, like the one you saw in the picture, when you leave it to keep
Continues, yeah.
they become like when you pick the potato up, you can just crumble it in your hand.
Yeah.
It's totally dried out. Yeah.
I've seen potatoes like that somewhere.
So again, just a historically interesting type of an organism that sure looks like a fungus, but actually turns out to be genetically quite unique, something much closer to an algae than a fungus. Remember, algae are also, at least in some forms, algae are also microbes. Now, here's the group of fungi that really have a practical application in our lives, and that's the kind that we refer to as saprophytic fungi. So this you can think of as the word saprophytic is a big descriptive term for a big combined group of different species of fungi. And what brings them all together as a group is the fact that these fungi decompose dead things. So these are the fungi that are out in the world that are responsible for breaking down all of the dead material, all of the dead organic material in the world. And of course, we're very dependent upon them. If we didn't have them, that material persisted, it would be a terrible, catastrophe for us. We need things to degrade. And one of the organisms that helps degrade dead bodies, dead things, are the saprophytic fungi. These organisms are not generally pathogenic. So they're out there doing their work in the environment and they don't bother us for the most part, except there's always an exception, except in people who really truly do not have a functioning immune system. So somebody who is in the later stages of AIDS, for example, or someone who was born with a genetic defect in their immune system, those people can become infected with saprophytic fungi. But anybody who has a functioning immune system, even partially functioning, won't be bothered by these fungi. So
Is that why they tell people who are undergoing chemotherapy not to dig in the dirt?
it's not just fungi for them. There's a lot of microbes in soil. It's also, there's a lot of parasitic organisms in soil, and there are pathogenic bacteria in soil, and there's all kinds of stuff in soil that you and I don't even think about, right? Because our immune systems are great. But when you're getting chemotherapy, your immune system is really beaten down. And so they just don't want you to take any chances when you're getting chemotherapy. That's right. Yeah, I can remember when we were kids. I grew up in a big family, and when we were kids, we were outside all day playing in the dirt. I can't imagine how much dirt we ate when we were kids. It's amazing that we don't realize how much our immune system protects us. So on the bottom of this slide, I have the names of three groups of saprophytic fungi. These are not genus names, by the way. These are higher up in the taxonomy order of fungi. So this is not the name of a genus. It's a category of fungi. And you'll see it all three of these end in mycota. So there's a group called the zygomycota. There's a group called the ascomycota, and there's a group called the basidiomycota. The term asco translates as sac, and basidio translates as club. And they got these names. I'm not sure about zygo, that one I'm not familiar with, but they got these names based upon what they look like. So some of these fungi look like they have structures, grossly visible structures that resemble a sac, and others resemble a club or a bat, if you will. So when you look into these groups a little closer, you see fungi that we encounter, relatively frequently. There's a type of zygomycota called Rhizopus, and Rhizopus is one of the genuses that grows on breads. This is not the greenish type of mold that grows on bread. This is one of the black molds. So it can be a bit of a pest in the food world, because this particular type of fungus loves to grow on breads. There's a type of ascomycota that we use to make bread. The name of it is Saccharomyces, and the species name is Cerevisiae. You don't need to know these names or anything, but Saccharomyces you can actually buy in the grocery store, all dried out for you in a packet, and you can bring it home and you can make bread with it. You can make yeast breads with it. So baker's yeast and also brewer's yeast come from Saccharomyces. Brewers, meaning the people who make beer, use this yeast in their recipe as well. So again, the ascomycota, important in our food production. And then finally, a couple of examples from basidiomycota. The fungus that grows and produces penicillin for us, the genus name is Penicillium. That is one of the basidiomycota. And then there's another type called Aspergillus. Aspergillus likes to grow on certain types of grain. It especially likes to grow on peanuts. So people who make those types of products, food products, have to watch out for Aspergillus because it can ruin their crop. I wanted to point out too that anytime you see a genus name like Penicillium, followed by the letters SPP, this stands for species. So when someone's just talking about the whole genus Penicillium, or I should say writing about the whole genus of Penicillium, not one particular species, they'll put this notation down, SPP, and we read that as Penicillium species. So all species of Penicillium. If we want to talk about a very particular species, remember, we use the species name like Cerevisiae. We could not live on this planet without saprophytic fungi. They are critically important to us. They do that work for us. They break down carbon-containing life forms after those life forms die. They don't kill things outright, but once something alive has died, the saprophytic fungi will come in and degrade it, take it apart, break it down, and return it basically to the soil, return it to the earth in its most basic forms. So we're very dependent upon them. They are all over the natural world, and that's a good thing. We need them. So let's switch gears here away from the gigantic world of fungi, and let's go down and think about the microscopic fungi for a few minutes. Remember, there are two forms of microscopic fungi. There are the yeasts and there are the molds. The yeasts are single-celled and the molds are multicellular. So only the yeasts can be considered microbes. They are single-celled microscopic organisms, so they fit our definition for a microbe. Now, there are lots and lots and lots of different species of yeast, but they all look pretty similar under the microscope. They range from being round or spherical to being sort of oval and everything in between. You can see egg-shaped yeast, but generally a rounded or oval-shaped organism. They are found widely throughout the natural environment, just like most types of fungi are. And yeasts, as we have said before in here, yeasts are produced through asexual reproduction, through a process that we call budding. So what happens is one yeast cell can reproduce all by itself. There's no male and female involved. Well, this parent cell, as we call it, will copy itself, and it will let go the new cell as a little bud coming off of its surface. We call the new cell the daughter cell. It's very sexist. We refer to them all as daughter cells. Now, sometimes, and again, we saw this in lab, sometimes as yeast start to go through this budding process, this asexual reproduction, as the new yeast cells are made, they fail to sort of break away. They fail to fall off of their parent cell. And sometimes you'll get long chains of yeast cells produced. When that happens, they can look similar to hyphae under the microscope if you're not careful, if you don't look at them carefully. So we give a chain of yeast cells a name. We call it a pseudo hyphy. Pseudo meaning, sort of like a hyphae, but not really a hyphae. Remember, the hyphae are those branches that mold produces microscopically. On this slide, over on the right-hand side, you're looking at a picture that you've seen before. This was taken from the strawberry that I mashed up and we looked at under the microscope. This is taken using the 100x objective lens, so we're under oil here. And you can see some of the hyphae here. This is the mold form. And you can see some yeast cells very clearly. And in this slide anyway, in this view, this particular field of vision, you could call these pseudo hyphae because I have a whole chain of yeast cells that are being produced as these yeast reproduce asexually. Up on the left over here, we're actually looking under an electron microscope. So the cells are much larger. This is a scanning electron microscope image. And if you remember, when you use a scanning electron microscope, you get an image of the surface of the cell. So these are, you can just get a feeling that these are three-dimensional structures. This is probably the best one to look at because you can really see there's a parent cell right here, and it's creating a daughter cell right here. It's budding off of the parent cell. And this bud will continue to grow until it becomes the same size as that parent cell. You can see another little bud down here coming off of another parent cell. This is really an amazing image. This is actually kind of a famous image because on this parent cell right here, I think you can just make out these two little dots right here. This is actually some of the genetic material inside of the parent cell, and it has condensed down because it's getting ready to be divided so that some of it goes into the bud that is about to be made, and the rest of it stays back with the parent. So that's a pretty amazing image. Again, you wouldn't be able to see anything like that under a light microscope. And down here, this is just a drawing of this budding process. The artist is trying to show us that this parent cell sends its own material. It doubles its own material. It replicates its DNA, for example, and it gives some of that to the bud. And the bud will then break off in most circumstances and go on to grow to become another cell. Sometimes, though, when we see it, it does this. You get that pseudo hyphae forming. Remember, any organism that reproduces asexually like this, where you have one cell reproducing to make another cell, the daughter is going to be genetically identical to the parent. They have the exact same DNA. Very different from sexual reproduction, where the offspring have their own unique DNA. So just like all the rest of the fungi, yeasts are very diverse. And they're not only diverse in terms of their genetics and how they look, but they're also metabolically diverse. I mentioned earlier that some yeast are aerobes, just like us. They require oxygen in order to perform their metabolism. Some of them are anaerobic. They don't require oxygen. There's a large class of them that are considered facultative anaerobes. I want to introduce this term here. We're going to visit it again and again through the semester. When you hear the term facultative to describe microbes, specifically a microbe's metabolic capabilities, facultative means that they're able to do one type of metabolism under normal circumstances, but if the need arises, they can switch over and do another kind. So a facultative anaerobe uses oxygen if it's available. They're going to do aerobic, for example, cellular respiration to make their ATP. Remember, ATP is that little molecule that all cells use as an energy currency. And these facultative anaerobes, if there's oxygen available, they're going to metabolize just like any other aerobe, just like you and I do. But if there's no oxygen available, they can turn around and do something completely different. They can do anaerobic cellular respiration, and some of them can do fermentation. So, if you and I run out of oxygen, we die. But these yeast can just switch over and start doing another type of metabolic pathway, which is a pretty neat trick if you think about it. So they can survive in all kinds of different environments because they're able to do that. Fermentation is a really important trait that some yeast have because we can use it. We can use it industrially or commercially to make all kinds of products that we like. A lot of fermented foods, for example, fermented beverages, the alcohols that people like to drink, and even industrial processes that require certain chemicals. Those chemicals are produced through fermentation. So in order to get these things made, we have learned how to grow these yeast in huge quantities in industry. We actually grow them in these gigantic vats, and we use gallons and gallons of nutrient media for the yeast, and we keep them under a low oxygen in order to sort of push them into fermenting. And as long as you give them the carbohydrate that they like to ferment, they will do it for you. And that, again, we take advantage of in a commercial sense and make these products. We're going to talk at length about how fermentation happens in a couple of weeks, but I just wanted to introduce it today.
May I ask
That's
a
okay.
question about anaerobic cellular respiration?
Yes.
Specifically, fermentation is anaerobic cellular respiration, but not all anaerobic cellular respiration is fermentation.
So I don't want you to worry about this too much today.
Okay.
Because we are going to talk about it. But
okay.
For today, I want you to know anaerobic cellular respiration is a completely different process than fermentation is. That may blow you away because a lot of students come into micro thinking that those two things must be the same.
Nope, that makes sense. Thank you.
So 3 pathways that we'll learn about as we go forward. Good question. All right. I want to spend a couple minutes now and talk about the other kind of microscopic fungi, and that's the molds. Remember, molds are not microbes because molds are multicellular. So they just don't fit our definition for microbes. Because they're multicellular, they grow as those branched structures that we call hyphae. The term that we use is filamentous. They grow in these long filaments or branches. The filaments look very different when you look from species to species of mold. There is some terminology I want us to be familiar with. The branched filamentous strands just generally are called hyphae. Now, the singular form of this word, you would just take the E off. So it would be hypha. And if you want to make it plural, you put the E back on and it's hyphae. Some mold grow what we call septate hyphae. And what that means is each cell is separated from the next cell by a wall structure that we call a septum. So when we look at the hyphae under the microscope, you can see where each cell ends and the next cell begins, because there's a wall structure between them. There's another kind of hyphae, though, called Pseudo Hyphy. That's a terrible word to spell. Pseudo Hyphy don't have that septum structure between the individual cells. So when you look at the hyphae, it looks like one big long cell. The difference is that you can see multiple nuclei in it. You can't tell really where one cell ends and the next cell begins, but every so often there's a new nucleus sitting there. And that tells you you're now into another cell. Now, here's a couple of sort of practical things to know about fungi. About molds. The vegetative form, again, the cellular form of hyphae can grow in or on growth media, nutrient media in the microbiology lab. So you can grow them in broth without any trouble, or you can grow them on the surface of an agar plate, just like we do with bacteria. Now, there's also a hyphal form that is purely reproductive. And here's how you know. Any hyphae that grow up above the surface of, say, agar, those are reproductive structures. So if you think through your own experiences, you've probably seen mold growing in certain circumstances up above the surface of whatever it's growing on. That's when it tends to look very fuzzy and cottony. It has now produced reproductive structures. When it grows close to the surface, when it grows right on the surface of something, that is just its vegetative form. What you're looking at there is essentially a colony. So yes, you can grow colonies of fungus on agar, just like you can grow colonies of bacteria on agar. And the tricky thing is you can't look at the agar plate and know for sure whether it's a bacterial colony or a fungal colony. It will trick you. Some of the fungal colonies look just like bacteria. They're perfectly round, they're smooth, they don't look fuzzy at all, they have no color. And some bacterial colonies will trick you into thinking they're fungi because they grow in very irregular shapes. So both bacteria and fungi can grow as colonies on agar and also on food. When the fungi start to grow up above the surface of the agar or above the surface of food, that structure is for reproduction. So when mold want to reproduce, they send these reproductive hyphae up above the surface. And those reproductive hyphae are where the reproductive spores are going to come from in some species. Remember, these are not like bacterial spores. They're not produced because the cell is stressed. They're produced because the organism is trying to reproduce. On this slide, you've got a couple of drawings over here on the left, and then you've got a couple of microscopic images over here on the right. So if you look on the left first, what we see drawn is the difference between first of all, these are both molds. They're drawings of molds. And the top one represents Septate Hyphae, and the bottom one is the Pseudo Hyphae. So if you look at the top, you can see this is a multicellular organism. This is a mold. It's microscopic, but it's multicellular. You can see where one cell ends and the next one begins because there's a septum in between each cell. You can also see they've drawn in blue where the nucleus is inside of that cell. Okay, this is a eukaryotic organism, so it's going to have organelles, including a nucleus. Now, down at the bottom here, you can't tell where one cell ends and one begins. You can see that suddenly there's going to be another nucleus appearing. So that's one clue that you're now into another cell, but there's no dividing line between the cells. This is the Pseudo type. Some species grow with Septate Hyphae, and some species grow with the Pseudo Hyphae. This is what they look like under the microscope. These images are taken under the 100x lens, so we get a nice high magnification. You can see right here on this purple branch, right here, this hyphae, you can see a division between the cells. You can see a septum that forms between one cell and the next cell. This is one organism right here. It's just that it has many cells in it, just like you and I have many cells. And each one of the cells is divided from the next by a septum. Now, if you look down here, this one has been stained differently. But look at this structure right here. This is also a hypha. It extends down here and down here. But if you look, there's no clear septum anywhere. It's just one long branch, but it has 1, 2, 3 lightly colored nuclei in it. So a little bit of anatomy about mold. Now, here's where molds get even more interesting, as far as I'm concerned. Mold can grow from a microscopic form into a macroscopic form. So the same species of mold can grow from being microscopic. It just gets larger and larger and larger until it's macroscopic, until it's visible with the naked eye. That's why you can see mold that's growing in your bathroom. That's why you can see the mold growing in the grout cracks. It started off being invisible to you as a microscopic organism, but it just got bigger and bigger and bigger until it became visible. Now, when mold becomes microscopic, all of those filamentous strands can just keep growing and growing and growing and growing. And this is what we refer to as a mycelium. So remember, we said when it comes to the macroscopic forms that fungi can take, out in the environment, mycelia are growing all over the place under the ground. Big, huge, enormous organisms that are made out of just thousands and thousands of filamentous strands. You may have heard there's a mycelium that's growing out in the West in our country. It's either Colorado or Washington State. And they figure this one organism is about 3.5 miles wide. And the way they know that is because they can go into the soil and they can grab a little bit of it. It looks white. They can grab a little bit of it, and then they can go 3.5 miles away, go in the soil, grab a little more of it. And they can take both of those samples into the lab and check to see if they are genetically identical. And they are. So this one organism, this one fungus growing as a mycelium is living, I think, in a state forest. I want to say it's Colorado. But yeah, they're all over the place. We don't think about them. We don't see them because they're under our feet. So remember, fungi are very diverse. We have to take a lot of steps in order to identify the exact genus and species anytime we find a fungal organism. You can, as I said, grow some of them into colonies, and they're going to have their own colony morphology. Fungi that grow as molds are going to have hyphal morphology that's unique to them. We can look at their reproductive spores under the microscope. They're going to have their own morphology. Remember, that word morphology just means body form. It's the way something looks. Now, here's something I do want you to be familiar with. The most modern way for us to identify a fungus is by taking that fungus into the laboratory and taking out some of its rRNA. Remember, rRNA stands for ribosomal RNA. So you have to harvest some of the ribosomes inside the fungal cell, and it's a little bit complex, but it's not as hard as it sounds. And what we do is we isolate the ribosomal RNA out of the ribosomes, and we sequence it. We determine the order of the bases in the RNA molecule. Every different species has a different sequence to its rRNA. So we can use this almost like a barcoding system. We can use this to identify different types of molds now and other types of fungus as well. Now, something else I should mention here. There are some species of fungi that will grow only as yeast. Some will grow only as molds. Some, as we have seen, can grow as both yeast and molds. It's very diverse. Any fungal organism that can grow as either a yeast or a mold, we give a name. We call that a dimorphic fungus. Di meaning two, and morph again meaning body form. So two body forms, yeasts and molds. Now we're going to switch gears a little bit here for the rest of our time together and take a look at a little demonstration that I put together for you. One of the things we need to know clinically about the fungi are that they are capable of causing disease in humans and other animals and plants, as a matter of fact. So there are fungal pathogens. But remember, the most common pathogens in humans are bacteria and viruses. So the question is, why don't we see more disease caused by fungi? And the answer is they are just not as able to evade our immune system. So when you see fungal disease in a human being, it's either secondary to some other disease going on, perhaps a disruption somewhere in the person's microbiome, or it's in a person who's just immune compromised. Remember, I mentioned somebody who's suffering from AIDS. Think about a burn patient who has lost some portion of their skin. They're quite vulnerable to fungal infections. People who have a disruption in their microbiome, maybe they have ulcerative colitis, or they have some other form of intestinal disease, and they don't have a healthy microbiome. The fungus can sort of take advantage of those situations and start growing. So we do often see them causing disease when something else is also happening, and the person's immune system is just overwhelmed, basically. You can imagine that in the laboratory, we have to be careful of microbial fungi. The spores, the reproductive spores, can get into the air quite easily, as can yeast. If we want to grow fungus in the lab, if we want to purposefully grow them in the lab, there's a special kind of media for them. I have the name of it right here. This, it's a strange looking word, but it's pronounced Sabarodes agar. And this agar, the agar that we grow them on has had nutrients added to it that are very specific. We put in a very short list of nutrients, including things like glucose and that peptone material we talked about. Remember, peptone is sort of partially digested protein. But here's the key part. We also put a very low pH in that media. So it has a pH of like 4 or 5. It's low by microbiology standards anyway. This will inhibit bacteria from growing on Sabarodes. So we have much less of a chance of getting bacterial contamination when we use Sabarodes. Only fungus is going to grow well on Sabarodes agar. So take a look at this image. Again, I'm not really set up here at home to do much culturing for you, but there are some tricks I can use, and this is one of them. You're looking at a baking dish here with some canned pears in it. You may have had canned pears yourself before. You can buy them in a heavy sugary syrup, but you can also buy them just in juice, and that's what I have here. Now, what I did was I emptied the can into this baking dish, and then I basically set it on the counter for about a week, undisturbed. It was in a dark area, and it was plenty warm. And as you can see, within about a week, I had colonies of fungus growing on these pears. Remember, bacteria might have trouble growing on these pears, right? These pears are sweet, and the water in the pear is bound up by that sugar, by that carbohydrate. The fungi, that doesn't bother them, but it bothers bacteria a lot. They're just not able to grow well on sweet foods like fruit. So this was, maybe 5 or 7 days along. And then you can see up close, you can see I've not got one species of fungus growing, do I? I have several different ones. How do I know? Because I can see different types of colonies growing. Remember, it's possible for 2 different organisms, whether we're talking about fungi or bacteria, it's possible for 2 different species to have the same looking colony. But if I see 2 different looking colonies, I know I have at least 2 different organisms. In other words, the way a colony looks is a feature of that organism. So every time it grows, it's going to look the same. So yes, if I see 2 identical colonies, this could be one organism, or this could be 2 different organisms that just have the same colony morphology. But obviously, this is a different one right here. It just looks very different, different color. Now, not to gross you out, but this is what the dish looks like after 2 weeks on the counter. I actually had this well covered, believe me, so none of this would contaminate the rest of my kitchen. Now, what we're looking at is not only some colonies growing on the surface of the fruit, but also all of this white fuzz growing up off the surface. These are reproductive structures now. So all of the fungus that managed to grow on those pears is now trying to reproduce. So it's sending up these reproductive branches, these reproductive hyphae that have little spores on the ends, and it's hoping that those spores will create new fungal cells. On this slide, I focused in on the corner of one of these pears. These are the same colonies that we saw at week 1, It's just that now they've gotten much bigger. And the reason I put this picture in is because here's a great example of what some people might look at and go, oh, black mold, right? It looks black. It has a black color. Remember, there is a genus of black mold that is very, very toxic. It's called Stachybotrys. I have the name down at the bottom of the slide for you if you're interested. But there are many types of mold that grow as black. So just because you see a black fungus growing, a black mold growing, you don't have to assume it's that black mold. You can see some green mold here. This might be Penicillium. Of course, I would have to look at it under the microscope to be sure. But all of this white fuzz that's growing up off the surface of the pears, these are reproductive hyphae. Just a little practical tip here. When you're growing fungi in the lab and you want to take a look, especially at the reproductive hyphae, the cotton ball looking hyphae, it's really hard to get a sample of them. If you tried to sort of just grab some of this, it would collapse almost like cotton candy in your hands. So a little trick that we use in the lab is we make a little loop of Scotch tape, and the sticky side is on the outside of the loop here. And what you do is you just touch the tape to the hyphal sort of clouds here, and you'll catch some of the hyphae on the tape. And then you'll take this tape and you just press it down onto a microscope slide, and you will leave some of those hyphae behind. It's essentially making a smear, but you're doing it with a piece of tape. You'd then have to heat fix that slide and then stain it in order to see them with any clarity. But it's just a little tip for sampling reproductive hyphae. Over here, I made a Sabrode plate just for fun. I grew a couple of colonies of fungus here. And then I took some of the reproductive hyphae and I gave the guy a smile. If you're not familiar, I'll try to remember this for one of our labs, but there's a National Society of Microbiologists, and there's a sort of a subgroup of all of the laboratory technicians who work in microbiology labs, sort of like a Facebook group, but more technical. Anyway, every year, these technicians have a contest where they try to grow organisms, microbes, on agar plates in designs. They try to purposefully grow bacteria and fungi and different species with different colors and different shapes. And they literally have a contest. It's an international contest to see who can make the most artistic agar plate. I'll try to remember to post some of the pictures from last year's contest so you can see them. The last thing I want to show you today before I let you go is a clinical application. So something where we deal with fungus in the healthcare setting, in a clinical setting. Remember, only yeast is microbial. The only fungal microbe is a yeast. There are lots of different species of yeast, but the only thing we're going to see in terms of microbial growth in a human regarding fungus is a yeast form. So over on the left side of this slide, you can see a Sabarode's agar plate, and it's got some colonies growing on it, doesn't it? These are strange looking colonies, to be sure. They don't look like those little circles that we looked at in our last lab together. But even though they look irregular and there's several different sizes, I could not look at this plate and say for sure that these are fungi. These could be bacterial colonies. Now, once I'm told that they're growing on sabarodes, now I can be pretty sure they're fungi, because bacteria just can't grow on sabarodes agar very well. So we took a little bit of this material. Oh, first of all, the plate was made from a patient sample, from some body fluid out of a patient. I think it was originally um, fluid from the bronchial tubes. So they did a procedure called a tracheal wash on this patient, as horrible as that sounds. And they collected some of the mucus and the material that this person had in their lower trachea, the tube that leads down into the lungs. And they smeared that material onto this agar and they grew these colonies. Now, when a sample was taken and a smear was made and stained, this is what they saw. Now, this image was taken not under the 100x lens. This image was taken underneath the 20x objective lens. We don't see many 20x images, do we? Because you can't see bacteria at 20x. We have to be at least 40x to see bacteria. These are yeast. These are yeast. Now, one of the features of yeast that makes them a little strange is that unlike bacteria, when you look around this slide, they're different sizes. They're different sizes. There are some that are really little. There are some that are really big. Some of them stained sort of lightly. Some of them stained really darkly. Remember how yeast reproduces. It buds. So these smaller cells are probably younger. They just haven't finished growing yet. And these larger and darker cells are all mature. So different, very different looking from a smear of bacteria. It turned out that this was a case of a very common type of yeast, but a very uncommon species. So the common type of yeast, in fact, the most common genus of yeast in human beings and other animals is a type called Candida. So this is a genus name, which is why it has a capital C, Candida. There are many different species of Candida. And again, in humans, most of the time, we don't think of Candida as being a particularly bad pathogen. It's not. It tends to be opportunistic. It tends to only cause problems in people who have other things going on as well. But this is an organism called Candida auris. I have the species name written down here. This is an emerging pathogen, not only here in the United States, but around the world. This is a species of yeast that is evolving to become a dangerous pathogen. This type of Candida, unlike all the other types, has been shown to cause primary disease in some people. In other words, they don't have to have some other problem going on. So we're very concerned about this in the public health world. This is a pathogen that, for example, the CDC is paying very close attention to. The WHO, the World Health Organization, is monitoring these infections around the world because this is a pathogen that the likes of which we really haven't seen before. And it's capable of causing some bad disease, things like pneumonia and so on. We're very concerned. So, you know, when I say, so Candida is the most common genus of yeast that causes infections in people, but it's secondary infections. One of the greatest examples for women, of course, is it causes yeast infections, vaginal yeast infections. And they're almost, well, I can't say almost always, but they are often a secondary type of infection. In other words, the woman has something else going on that causes an opportunity for the Candida to establish itself and grow in the vagina. One of the most common causes of a vaginal yeast infection with Candida is antibiotic therapy. If you've ever had to take an antibiotic because you have a bacterial infection somewhere, let's say you have strep throat or you have some other type of infection caused by bacteria, when you take the antibiotic, it's only killing the bacteria. It's killing the bacteria. The problem is that it's not just killing the bad bacteria. It's also killing lots of other good bacteria that is part of your microbiome, including the microbiome bacteria that lives in your vagina. And what happens is, as those good bacteria die, the yeast take advantage and they start to grow and fill up that empty space, that empty real estate that the bacteria used to occupy. So it's very, very common for women to develop a vaginal yeast infection after they've been on antibiotic treatment. And it's confusing for some women because they think, well, I'm taking an antibiotic. Why doesn't the antibiotic kill the yeast? But of course, antibiotics, or I should say antibacterial drugs, only treat bacterial infections. You have to take a different kind of drug, an antifungal drug, to kill yeast. So yeah, even for, like I said, a burn patient has lost a barrier to infection. They've lost part of their skin, and the yeast will take advantage of that. And they will try to grow, and they're often successful at growing on those burn wounds because the body is fighting off that other problem. It's your immune system is overwhelmed from a severe burn. So yeah, it's very interesting. They are opportunistic pathogens. They like to take advantage when something else is going on, when something else is wrong. Any questions about any of what we covered today? Any questions about any of the terminology? Again, we just touched on some of the taxonomy of fungi, but it's really the microbial, the yeast that apply to us in microbiology.
I do have a question. I'm sorry. I keep asking questions.
No,
but I'm
that's all right.
fascinated.
That's all right. I appreciate the questions.
So when you think of older adults whose immune systems are often compromised, does that mean that like fungal infections are more prevalent in
That's a great question.
in older patients, and particularly in a long-term care or medical setting?
That's a great question. So just, we associate advancing age with sort of a breakdown of a lot of the immune protections in the body. And it is true, it does happen. But I'll say that the healthier elderly, in other words, those that are tending to be living independently and are still mobile and are still out and about, going grocery shopping and walking and things like that, even, into their 80s and 90s, don't tend to get fungal infections. It's the people, like you said, who are really becoming compromised. It's the people who are bedbound, the people who are in long-term care facilities and are not mobile, people who are disabled and elderly. They're in wheelchairs. People who have to have chronic medical treatment. So I'm thinking about people who are routinely catheterized, people who maybe have to have a central line in because they have other health problems going on, people who are elderly and on dialysis. Those are the folks you got to watch for. They're more able to pick up a fungal infection than just another sort of healthy elderly person. Yeah. Fungi are tricky. They, like I said, they're opportunistic. And if somebody gets really beaten down, medically speaking, and they see an opening, they'll take it. The other group that we're learning more and more about in recent years are folks who are chronically, so years and years, living with a really poor microbiome. I'm thinking about right now in my head, I'm thinking about some research I read about people who are chronically morbidly obese and they have really poor diets. Their microbiome gets to be in really bad shape over time. Their normal healthy bacteria in their gut tends to die off because it's being fed really poor quality food in some of these people, not all of them, obviously. And what happens over time is, again, the body is so beaten down, the immune system is just beaten down by this person who is not only morbidly obese, but also has a really unhealthy microbiome. The yeast infections can start to show up. You'll start to see these people getting intestinal infections, and you'll start to see them getting more and more skin infections, vaginal infections. They get yeast infections in the folds of their skin. And it's because their whole body is just chronically unhealthy. Yeah. But yeah, we have to be aware of it in long-term care facilities. We have to always be on the lookout for these really compromised folks who are vulnerable to opportunistic infection. Good question. One of the ways, historically, one of the ways that we knew back in the 1980s when we didn't know about HIV yet, when HIV was first emerging, one of the ways we knew something was wrong, that something was really wrong, was because patients were coming in to the clinic with fungal infections. Young men, young gay men were showing up in clinics with what's called thrush. It's a fungal infection in the mouth. Or they were showing up with fungal infections in their lungs. And the doctors and the scientists were like, what is this? These are young, healthy men. They shouldn't be getting fungal infections. And of course, it turned out that these poor men had been infected with HIV and had now developed AIDS. And their immune system was just not working. And the fungus took advantage and moved right in and started growing. So yeah, they are definitely opportunistic. Any other questions? All right, ladies, I'm going to let you go. Remember, if you haven't already, make sure you get your lecture exam into me. And keep up with your other lecture work and quizzes and so on. And I will see you next time. Look for that short video that I'll post for you about the colony morphology. Watch for that in our playlist. I'll let you know. I'll send an announcement out too when I post that. All right. All right. Have a good evening.
Thank you so much. You too.
I'll see you next time.
Bye-bye.
Bye-bye.